Well, what's really unique about the tetramer assay is that it allows you to directly visualize antigen-specific T cells. The tetramer is the main unique aspect of our assay. That's coupled with an amplification step that we do. We isolate the T cells and then we grow them in an environment that enriches the sample for the T cells that we're interested in. Then we're able to specifically label just those cells and visualize them on a flow cytometer. And so with previous assays, things like CSFE or proliferation assays that have been around for decades, what you're getting is just kind of an overall bulk measurement of what's going on with the whole T cell population. But you've got millions of T cells in the body, and they see a whole diverse set of patterns. If you're lucky, maybe 1 in 200,000 to 300,000 of the cells in the sample will be the one that you're looking at. And that's why this assay, it does that enrichment steps. And then with a tetramer, because you're directly staining, you can actually visualize away from background a population that's less than 1% of the total number of cells. And so you can see very, very small differences, and you can get a fairly precise count of the number of T cells that are in that blood sample, that are in that individual, that recognize your pattern of interest. But it doesn't stop there, because the tetramers also allow you to actually not only label those T cells, but isolate them for further analysis.
And the tetramers that you use are actually made here at BRI, is that correct?
Yes, we're one of a very small number of labs in the country that have successfully made Class II tetramers. Our intent is to distribute these reagents at cost to any laboratories that are interested in importing this technology.
What would you say are the most difficult or challenging aspects of this assay when it comes to replicating it in other labs?
Well, as I think about it, there's really two. The growth of the T cells is something that really is a bit of an art. You have almost daily decisions as far as whether the cells need additional nutrients or additional space to grow. And then the second part that I think is probably the most difficult for an inexperienced operator is actually the data analysis, kind of figuring out which part of the signal really is a positive response versus which part is just inherent to physics and attraction, and the fact that everything has some degree of stickiness to it is something that, again, takes a lot of experience, especially in contexts where the overall response is going to be a little bit weak.
How does the information you'll generate with this assay have potential to benefit patients?
At this point, one of the first hallmarks that comes up in any autoimmune patient is kind of a constellation of antibodies that are specific for that disease. We know from the fundamentals of immunology that wherever there's an antibody response, there's always a T cell response first. And so if we could look in at-risk individuals and see that jump in T cell activity that would precede antibody formation, that would then help us kind of define really a new level of risk, where individuals are headed towards having autoantibodies, which then hopefully would open up a window for interventive therapies. This procedure will purify and expand antigen-specific CD4 positive T cells and visualize them using tetramers. Reagents and equipment. Class II laminar flow biosafety cabinet. This will be the primary workspace for the procedure. 50 milliliter conical tubes, primary tube for PBMC isolation. 1x PBS. This should be calcium and magnesium free. Phycalpac Plus. Foil wrapped to protect from light. Drummond pipette aid. CoStar 10 ml pipette. Aerosol canisters with 50 ml inserts used to prevent blood contamination of centrifuge. GS6R centrifuge. Transfer pipettes. Pasteur pipettes. Hemolytic buffer containing 8.3 grams per liter ammonium chloride, 1.0 grams per liter sodium bicarbonate, 0.04 grams per liter disodium EDTA. Trypan Blue, 0.2% solution in PBS. Hemocytometer. 15 milliliter conical tube. Running buffer containing PBS supplemented with 2 millimolar EDTA plus 5 grams per liter BSA. Max CD4 Isolation Kit 2, purchased from Miltenyi Biotech. EasySep Magnet Block, purchased from Stem Cell Technologies. Other magnetic cell enrichment platforms could be used with equal efficacy. 5 milliliter polypropylene tube. RPMI 1640, supplemented with 25 millimolar HEPEs, used to make T cell medium. Pooled human serum, used to supplement T cell medium. Penstrip, used to supplement T cell medium. 500 milliliter 0.22 micron bottle top filter, sterile. 48 well plate, used for in vitro expansion culture step. 37-degree CO2 incubator. 20 milligram per milliliter solution of synthetic peptide containing the epitope of interest. 5 milliliter polystyrene fax tube. Peptide-loaded class 2 tetramer used to visualize antigen-specific T cells. Anti-CD3, anti-CD4, and anti-CD25 antibodies used for co-staining antigen-specific T cells. Fax caliber flow cytometer, or equivalent. Before starting the procedure, assemble all materials and equipment in your workspace. The following demonstration will cover the following steps: PBMC isolation, CD4 positive T cell separation, in vitro expansion culture, visualizing T cells by tetramer staining. Step 1. PBMC isolation. Obtain a blood sample. Blood should be collected in syringes or blood tubes and anticoagulated with heparin at a 1:50 ratio to prevent clotting. Expect a yield of about 1 million PBMC per milliliter of blood, about 40% of which will be CD4 positive T cells. Aliquot the blood into 50 milliliter conical tubes, 25 milliliters of blood per tube. Gently mix the blood before aliquoting to distribute the plasma. Add PBS, bringing the total volume to 40 milliliters, and underlay by drawing up 11 milliliters of Ficol, inserting into blood tube, and carefully removing the pipette aid from the pipette. The Ficol will slowly drain into the bottom of the tube. Once the level has equalized, slowly remove the pipette from the blood tube and discard. Cap the blood tubes and move into the aerosol canisters. Firmly close the canisters and centrifuge at 900 times G for 20 minutes, with no break. It is critical that no break is applied at the end of this spin, as the braking force will disturb the layer of cells. After the spin, the PBMCs should form a distinct white layer between the yellow plasma above and the transparent Ficol below. Gently skim off white blood cell layer using a transfer pipette and transfer to new tube. Some plasma and Ficol may be drawn up with the PBMCs, but avoid drawing up any of the red cell layer. Typically, 2 blood tubes can be combined into 1 cell tube at this step to increase the pellet size. Add PBS, bringing the total volume to 50 milliliters, and centrifuge at 500 times G for 15 minutes on low break in aerosol canisters. Aspirate liquid using a Pasteur pipette, taking care not to disturb the pellet. Treat with hemolytic buffer by adding 5 to 6 milliliters to each tube and gently mixing to remove clumps. Incubate for no more than 5 minutes. Add PBS, bringing the total volume to 50 milliliters, and centrifuge at 230 times G for 10 minutes on low break. Aerosol canisters are no longer needed for these slower spins. Wash 2 times. Aspirate liquid using a Pasteur pipette. Fill tube with PBS and centrifuge at 230 times G for 10 minutes. Prior to the final wash step, remove an aliquot of resuspended cells, dilute with trypan blue, and count using a hemocytometer. This cell count will dictate the reagent volumes used in the T cell separation step. Step 2. CD4 positive T cell separation. Aspirate liquid and add running buffer to bring the total volume up to 40 microliters per 10 million cells. Usually this requires 30 microliters of buffer plus the residual pellet volume. Transfer this volume to a 15 milliliter conical tube. Add antibody cocktail from the CD4 isolation kit, 10 microliters per 10 million cells. Cap tube and place on ice for 10 minutes. Remove tube from ice, remove cap, and add 30 microliters of running buffer per 10 million cells. Add magnetic beads from the CD4 isolation kit, 20 microliters per 10 million cells. Cap tube and place on ice for 15 minutes. Add 10 volumes of running buffer to wash and centrifuge at 230 times G for 10 minutes. During the spin, set up the magnet and 5 milliliter polypropylene tube in your workspace. Aspirate liquid from cell pellet. Add 2 milliliters running buffer and remove clumps using a transfer pipette. Using the same transfer pipette, transfer cells into the 5 milliliter tube and incubate in the magnet for 15 minutes. Carefully decant the CD4 positive T cell fraction into a marked 15 ml tube by inverting the magnet and emptying all but the last drop. Add another 2 milliliters of running buffer to the 5 milliliter tube and remove from the magnet. Gently rinse the CD4 negative cells off the sides of the tube and transfer to a marked 15 ml tube. Add 2 milliliters T cell medium to each tube. Remove aliquots for cell counting and centrifuge at 230 times G for 10 minutes. Dilute cell aliquots with trypan blue and count using a hemocytometer. These cell counts will dictate the number of wells used for the expansion culture step. Step 3. In vitro expansion culture. Aspirate liquid from the CD4 positive and CD4 negative cell pellets. Based on the cell counts, add media to the CD4 positive cells to adjust the volume to 3 million cells per mil. Add media to the CD4 negative cells to adjust the volume to 10 million cells per mil. The expansion culture requires 3 million CD4 negative cells per well and 2 to 3 million CD4 positive cells per well. Typically, CD4 positive cells are the limiting population. Aliquot CD4 negative cells into the appropriate number of wells on a 48 well plate by adding 300 microliters to each. Place the plate in a 37-degree incubator for 1 hour. Remove the plate from the incubator. Wash the wells by adding 500 microliters of fresh media and gently pipetting up and down 12 to 20 times using a transfer pipette and removing all of the liquid. This wash will leave behind a layer of adherent antigen-presenting cells. When the washing is complete, add just enough media to wet the bottom of each well, roughly 100 microliters. Otherwise, the adherent cells will dry out. Add 2 to 3 million CD4 positive cells to each well. If necessary, add additional media to bring the total volume in each well up to 1 milliliter. Add the desired peptide to each well. The typical concentration for stimulation is 10 micrograms per milliliter final. Place plate in a 37-degree incubator for 7 days. After 7 days, add 50 microliters of hemogen IL-2 to each well. On each subsequent day, monitor cell growth using a microscope. Feed wells that are not yet confluent by removing half of the media, adding fresh media, and 50 microliters of IL-2. Split confluent wells by resuspending the cells, moving half of the cells to an empty well, adding fresh media, and 50 microliters of IL-2. Return cells to incubator. The expanding cells will be ready for tetramer staining after 13 to 15 days of culture. Step 4. Visualizing T cells by tetramer staining. Purchase or assemble tetramers to match the peptide MHC combinations used to stimulate the CD4 positive T cells. Remove plate from incubator. Carefully draw off half of the media from each well. Transfer an aliquot of cells from each well, typically 75 microliters, or about 50,000 cells, into a 5 milliliter polystyrene fax tube. Add tetramer, typically 1 microliter per 50 microliters of total volume, and place in 37-degree incubator for 1 hour. It is also advisable to stain a second aliquot of each well with a mismatched tetramer as a negative control. Label cells with anti-CD3, anti-CD4, and anti-CD25 antibodies. Additional or alternative antibody markers can be used. However, do not use PE-labeled antibodies, since this channel must be reserved for the tetramers. At this time, also label any single color or isotype controls using extra cells. Incubate antibody-labeled cells for 15 to 30 minutes at 4 degrees. Wash each tube with 0.5 to 1 milliliter of running buffer and centrifuge at 230 times G for 10 minutes. Carefully decant liquid from the fax tubes. Calibrate flow cytometer using reference beads, single color control tubes, isotype controls, etc. Analyze each tube using the flow cytometer. The tetramer-positive cells should be a distinct population among the expanded CD4 positive T cells. Tetramer-positive cells are usually CD25 positive and often CD4 high.